All right, good morning. I'm kind of missing that summer-like weather. It's freezing out there. My asparagus froze. I'll have to eat it all. What I'm going to do today, this is for the lab. All right, I'll send a little announcement out, but I'm going to give you sort of we'll call them two big lab quizzes. They're not going to be too bad, but we'll do it in two parts. The first one will be the respiratory system and blood cell identification. That's it. It'll probably be, 50 questions or less. They will be, for the most part, multiple choice. You'll see a picture, and then there'll be like 10 options underneath. You pick the appropriate one. So again, it'll be easier than what I'd have done to you in the lab, but we'll make do. So white blood cell identification, or blood cell identification in general, red blood cells aren't going to be hard, and the respiratory system. On the respiratory system, it'll be a lot of the models, which I put some pictures up. I'll mention that in the announcement. It'll be the models from our lab, and it will be human cadavers, which I have a few pictures up for those as well. No point using cats since it's human anatomy, and we use those in a model because they're easy to get, but you're not in the lab, and there's no reason to use cats. So it'll be, the human models I've got photos of them already posted, and then a few pictures of cadavers. Now what I'm going to go over today, the blood cell identification. Normally do this in lab. What I'm going to do is kind of give you a quick outline on the whiteboard here, how to identify them, and then I'll go through the little PowerPoint that I have with the actual pictures. They're micrographs of the slides we normally use in lab. Now, mentioned blood already in lecture. Of course, it's a connective tissue. 55% or so of its volume is plasma. We'll ignore that. And then it's got some cell components, what they call formed elements. These are what you have to identify. There's only like 7, so it's not too bad. All right, so the formed elements will include, I'll go way up here. I'll just go R, B, C. These are red blood cells. They'll fill most of the slide. 99% of the cells or so are red blood cells. They're easy. They're kind of pinkish, reddish looking things. They're described as biconcave discs, kind of like a donut that didn't make it. There's bunches of them. You missed that, I'm going to mark it wrong twice because they're just easy. So there'll be a lot of red blood cells. No nucleus in them, nothing else. Again, when red blood cells are formed, when they're released into the circulation, most of the organelles are ejected. No mitochondria, no nucleus. Basically, as I've mentioned, bags of hemoglobin more or less is what they are. So those will not be a problem. The ones that are a little difficult are the white blood cells. All right, the white blood cells are part of your infection fighting system. There's about 5 types you'll have to identify. They're separated into 2 groups. Now on here, I'm going to kind of just describe how you tell them apart. All the white blood cells are separated into 2 categories. There are one type of white blood cell is called a granulocyte. Now, the way you'll identify these things is based on the morphology, the characteristics of the nucleus, which will stay in a dark bluish black, and in the cytoplasm, there may be cytoplasmic granules, little inclusions, vesicles, that sort of thing. As you can guess, the granulocytes will have granules in the cytoplasm, depending on the prep and the magnification you're using. So the first one, the most common type of granulocyte is a neutrophil. Now, you're going to have to identify these, which really isn't too bad. Also, just give me a brief, 1 or 2 word function, perhaps, and their relative frequency. One of the things they do with white blood cells, they can use them diagnostically. Certain white blood cells play a role in fighting off different types of infections like bacterial, viral, parasitic. And so if they see these numbers shifting, it gives them some idea of what might be ailing you. So I'll give you a brief function and a description and their relative frequency. In a normal unsick person, if you count 100 white blood cells at random, about 60 of them will be neutrophils. This is what they call a differential white blood cell count. You do an absolute count number of cells per microliter, that sort of thing. But this is just, if you just randomly count 100 out of somebody's blood sample, how many are neutrophils? It would be about 60%. And neutrophils they're phagocytic. They'll engulf cell fragments, viral particles, bacteria, that sort of thing. And they tend to increase in number when you have an acute infection, early onset of an infection, bacterial or viral. The way you'll identify them, the nucleus of a neutrophil is multi-lobed. You see 3 or 4 kind of lobes of the nucleus, kind of like big beads on a string, and it can be twisted into any kind of configuration. So imagine this is a nucleus here. You've got 1, 2, 3 or so lobes, a little constriction in between. These are granulocytes, but on the magnification we use a 40 power objective lens, so we have a total of 400 magnifications. You can't really see the granules. They're so fine, they don't resolve with the systems we're using. So you won't see much in the cytoplasm. There's some really fine inclusions, that's about it. So that's the neutrophils, you'll see boatloads of them. The next one. An eosinophil. It's named because of its staining characteristic. Eosinophils, they have granules that have an affinity for a histological stain called eosine. That's where it gets its name from. These are quite distinctive. All right. They will have a bilobe nucleus. So it's kind of 2 big wads there, 2 big lobes, with you may see a little sort of isthmus, little strand connecting them. Bilobe nucleus, not multi-lobe like the neutrophil. And what usually makes these really distinctive, the cytoplasmic granules. They have very obvious, very large, depending on the prep, they'll be pinkish, orangish, or often reddish dots in the cytoplasm. The eosine stain is a reddish, yellowish, pinkish stain. That's why they're called eosinophils. The granules have an affinity for this pinkish stain. So those are eosinophils. Eosinophils, they'll occur about 2% of the time. So if you randomly count 100 white blood cells, you get about 2 eosinophils. There's a range, can't remember it off the top of my head. I don't know if my textbook's here, but it's around 2%. So they're not terribly common, but they're distinctive. When you see them, you'll know what they are. One of their roles is to fight off parasitic infections, particularly parasitic worms. So if you have a worm infection, you would expect to see eosinophil numbers go up. That's their main function. They have some impact on allergies as well, but that's not a real function, that's an abnormality. And then third, try and squeeze it over here. The third one, well, I had pictures I'll show you, so you'll be able to see one. If you're actually doing a scan through blood cells, you won't find very many of these. The third one is called a basophil. Basophils occur about 0.1% of the time. That means if you count 1,000 white blood cells, you're likely to find one. They're not very common, and it's really hard to find one that's actually got good characteristics that all the students can identify, but I'll have pictures, so it'll be okay. Now, what makes these distinctive? They also have a bilobe nucleus, like an eosinophil, but it often appears a little more compact. It's not as spread out. The lobes are sort of more combined. But what makes it striking, if you see a good one, basophils have very heavy, dark, bluish black granules that look like little black polka dots in the cytoplasm. Again, these granules have to be in the cytoplasm. The nucleus in many of these preps will also look granular. Nucleus doesn't count. You're looking for these dots in what's obviously the cytoplasm, and it'll be a heavy bluish black. Basophils, they go to a site of injury and they release some chemicals. They release histamine, which causes localized inflammation. It swells up because the capillaries get more permeable, gets a little red because the blood flow increases. And it also releases heparin, which is an anticoagulant to clear blood clots out of a damaged area. So that's basophils. All right, those are the granulocytes. The other type is called A. Granulocytes. They have a clear cytoplasm, no inclusion, no granules. There's only 2. The most common is called a lymphocyte. And again, I'll have them on pictures labeled, so these can be hard to read. I've watched some videos, it's a little tough, but lymphocyte. Lymphocytes are relatively small cells normally. How do you know how big they are? You got all the red blood cells around them. You can kind of use that to gauge relative size if you have to. Lymphocytes are not as big as some of the other white blood cells, kind of tiny. What makes them distinctive is the nucleus. It's a big round or oval nucleus, which actually takes up most of the cell's volume. All you might see is a little crescent of clear, kind of sky blue looking cytoplasm peeking off one side of the nucleus. The nucleus is huge. Lymphocytes, we've mentioned in lecture, they're the cells that produce antibodies, which they're talking about on the news and all that sort of thing. You get a new antigen, these lymphocytes will eventually start producing antibodies, which give you immunity. So if they're looking for now, or checking for immunity for the COVID-19, they're checking for antibodies. If you have them, it means you've been exposed to the virus. Might have some immunity. I don't think they've sorted that out yet. That's one type. The last, and these make up about 30% of the white blood cells, so lymphocytes are also very common. Last one is one of the bigger white blood cells. It's called a monocyte. They'll make up about 5% of the white blood cell volume. And it will tell you most of these white blood cells leave the circulatory system. Lymphocytes are phagocytic. That is, they engulf, foreign material, bacteria, dead cells, that sort of thing. They tend to increase in number when you have a long-term or chronic infection, but most of their activity is actually when they leave the circulatory system. They can move like an amoeba, and they squeeze between the endothelial cells, or they'll move to a site of injury. Once they're out of the circulatory system, they call them macrophages, like what I mentioned in the lungs. But in the circulatory system, there are monocytes and big cells, and if you have a good one, their nucleus is a big, heavy, kind of kidney bean-shaped nucleus. So it's big and fat. Even this one can be shaped like a U, but the neutrophils, it's a spindly little nucleus. Monocyte, it'll be a big, fat nucleus. The cytoplasm, nothing in it. It'll just be clear, kind of transparent, sky blue looking color. That is the monocyte. The only other components you might see scattered around in the blood smear will be some things that look like dots in most of our pictures. They're not very big. They're little tiny, usually stained purple, dots all over the place. Those are platelets. They're not complete cells. As they form, there's a big cell called a megakaryocyte that fragments when it's released. It's still membrane bound, but it's just a little fragment of a cell. These are the platelets. They play a role in hemostasis or blood clotting, coagulation. That's it. So you got 5 white blood cells, the platelets, red blood cells. You identify those. That's going to be part of the lab exam. Be able to list me the functions somewhere. I might ask them in the lecture lab. I'm not sure yet. But, just a brief function and the relative frequency or occurrence of these things, you'll be good. Now, what I'll tack on at the end, I'll go through the PowerPoint I've posted to look at actual micrographs so you can see what they really look like, not these ugly stick pictures. I'll be back. Alrighty, here is one of the PowerPoints on white blood cells. I'll actually probably post 2. This one had slightly better pictures. And this is a composite. There's 4 different photos here. This one up in the top left, that is a neutrophil. See the dark bluish black nuclei, and there's one lobe there, a little strand, another lobe, maybe another one. That's a neutrophil. All around it, these are the red blood cells. They'll be everywhere. That little tiny purple speck, you can't quite see it there. There's one over here. Those are the platelets. Adjust things here a wee bit. That looks better. Okay, over here, another neutrophil, a little heavy looking nucleus, but it's still got lobes. Again, platelets. Bottom left is a really cruddy picture of a basophil. I've got another better one later. And here's a lymphocyte. Again, fairly small cell. You see that large oval nucleus takes up most of the space. There's just a little bit of cytoplasm, that little crescent you can see right there. I'll put this one up next. Here's a couple more neutrophils. You can see the multi-lobe nucleus. This one
is a monocyte. Big, heavy nucleus, kind of bean-shaped if you use a little bit of imagination. Another one down here, that one's a nice bean-shaped nucleus right there. Cytoplasm is clear. Again, you can see a platelet there. I think there's another platelet over here. Here, this is an eosinophil. See, these are easy. There's that bilobe nucleus, and then you see also pink polka dots in it there. That's the cytoplasmic granules in an eosinophil. Again, they're only about 2% of the cells, and they'll, fight off parasitic infections, particularly worms. Another lymphocyte here. You see the heavy oval nucleus, a little bit of cytoplasm there. That one's too blurry. There's a monocyte. It's kind of odd looking, but it's a big cell, big heavy nucleus. It doesn't really look bean-shaped in this example, but it's a monocyte. Here's a few more. Now, this is the best picture I have of a basophil. There's a lobe of the nucleus. There's another lobe down here. And you'll see these dark bluish-black dots in the cytoplasm. That makes it a basophil. Releases histamine and heparin. Smallest frequency for this one, only about 0.1% will be basophils. Another lymphocyte. Here's a good eosinophil. Again, you see the bilobe nucleus, little red, pinkish polka dots. Neutrophil, a couple more neutrophils. There's another decent monocyte. And then the rest of this, if you want to go through it, it's like a little practice quiz. It'll have a blank here. At the end, it'll give you the answers. That one's actually a lymphocyte. But that's it. Those are the white blood cells, red blood cells, and platelets you'll need to identify. I'll post this up in a bit.